was growing up in Pittsburgh like? It was good. It was a nice place to grow up.
Yeah, could you please?
Small city though.
I think it's worth it.
Definitely happy to be somewhere a little bit larger now.
Just a small startup.
Yeah, I'd say it's like a generation. Small city. Kind of gets
It was a bit of a,
I mean,
it was
they
a
get
lot of
gets
energy at the beginning, but then it was
pretty quickly. That's fun to do.
kept up for a while. I mean, I think the wave,
But definitely good.
I mean, what was happening is that
If you want, even you can make it narrower. Like,
and then I think the
if you
wave
want.
was left, it was now.
Right, because it's.
But I think it would be.
As long as everything
I think we should.
Summertime or something.
Yeah, I don't know what how's usual.
This is on Zoom, but I guess it's not. They see this. They see, so they'll see like you as long as you stand over here and their slides. Yeah.
That's a good idea.
Yeah. You sort of spit it with those genomics.
And then you can get questions from there, right? At the end, you can. So we'll just have one. There's a question online.
Yeah.
Just think of some ideas. And I thought it was
I think it'll
helpful,
pop
right?
up here in the chat.
You know,
Oh, it's a chat, yeah.
You have a
Well, you would-
that subject.
Yeah.
Fei Chen, but he's done a lot of this with our staff.
Oh, right. Okay.
I talked with him about sequencing and things.
Good to get started. Nelson?
Yeah.
All right, thank you everyone so much for coming to today's GSI seminar. I'm really excited to have Fei Chen here. Fei did his PhD in bioengineering at MIT and then subsequently became a Schmidt Fellow at the Broad Institute and now leads his own group. And Fei is going to talk about some of the work that his group has been doing, specifically developing and applying some novel spatial omics tools. So take it away.
Thank you for the introduction. Thanks for having me. This is working all right? Okay. Yeah, so exactly, as I said, as Jason mentioned, I'm going to talk a lot about some technologies that my lab has been developing on really bridging the world of genomics and also tissues. And maybe just as a motivation for what that means is that I guess the last decade or more than a decade, maybe 15 years, 20 years or so, has seen this revolution in genomics technologies, and many of you guys may have also participated in. This is driven by this exponential decrease in sequencing costs. It has allowed us to sequence genomes, but more recently it has allowed us to sequence individual cells from cells and from tissues, and it's allowed us to start defining what cells are molecularly from their transcriptomes and also maybe epigenomically from their gene regulation perspective. But most of this was traditionally done in bulk, where you grind up your tissue, and more recently it's been done in single cells, where you kind of isolate single cells from tissues into like a slurry of cells. As you know, tissues are not composed of slurries of single cells. They're like some organization. Their function comes from the organization and interactions between cells. And the traditional lens through which we've looked at that has been through microscopy. For hundreds of years, we've looked into the lens of a microscope and seen, the organization of biology, as well as like what's going on, for example, when it's disorganized, or like in pathology, where you can associate disorganization of tissues with actual disease. And so the idea that we've been working on and the space that we've been trying to make an impact in is like trying to bridge these two worlds. Like, how do we collect genomic measurements directly from the context of tissues and still know where those measurements came from, and then how do we like understand the relationships in that context. And so we've been developing like technologies in two spaces, and I'll really only talk about one of them today, but I'm happy to talk offline about another. The first is like sequencing-based tools. These are like DNA and RNA sequencing-based tools that allow you to capture mRNA or DNA from cells and tissues and still maintain where those reads came from. That's like the basic idea. And the second class of things that I won't really talk about today, but I'll just give you a primer on is just like imaging-based tools, wherein like you can go on a microscope and you can start performing some like in situ sequencing, like sequencing reactions, combinatorial like fluorescence microscopy inside of cells to get a sense of where biomolecules are. For example, here's like a bunch of spots representing different reads from a nucleus, and then you can kind of reassemble the structure of the genome from that. But and then where are we applying these? We're trying to like basically trying to understand how tissues are organized in terms of molecularly defined cell types, and then also what's going on when there is pathology, both in the brain and in cancer. So just to orient you guys on the technology that we'll talk about today, we've been using this approach called SlideSeq, which we developed, and effectively, SlideSeq is a way to make like a high resolution capture array for nucleic acids. And how we do that is using these barcoded beads. Each one of these beads is a individually barcoded 10 micron. Maybe this will work. Okay, there we go. So each one of these beads is like a 10 micron polystyrene bead with millions of oligonucleotide sequences on it. And each one of these for sequences maybe they have poly-T to capture mRNA, and then uniquely they have this spatial bead barcode, which is different between beads, but clonal for each bead. So it's like a sequence barcode that identifies the bead. We deposit these beads randomly onto like a slide, hence the SlideSeq part. Each one of these circles is a 3 millimeter diameter array of about 100,000 of these beads. And here they are sitting on the surface. They like basically get spun onto this glass slide, and they're randomly deposited. So then what we do is we take a microscope and we actually go and perform the same sequencing reactions that you would do on a next generation sequencer, but we have some custom fluidics that allow us to sequence these beads. So A might be green, and C might be yellow, and red might be G, and this is T. So that's like, one base, and then we do that over like multiple bases. And then what you do is you assemble like this random, these beads are randomly deposited, but now you know the correspondence between their sequence and their XY locations. So you've made like this array of pixels effectively. And now instead of capturing photons, the idea is that you want these pixels to capture mRNA. And so when you do an experiment, what you actually do is you take a piece of tissue, fresh frozen tissue, and you section it, maybe a 10 micron section. You put it onto this array. The RNAs diffuse onto these beads, and they're locally captured, and then you amplify that library. When you amplify that library and you sequence the cDNAs, you know the RNA identity, but you also know the location. And I'll show you some data and how we're analyzing it in a second. But that's the concept. And then we're generally like interested in a couple of problems. One is like how are tissues organized with respect to like molecularly defined cell types, right? Now we have this molecular definition of what cell types are. How are tissues composed of those? And then the second two are kind of related. One is how do we understand, a cell type is kind of like a classification, but cells, gene expressions change as a function of their environment. So how do we understand, like within a cell, how does their gene expression change as a function of where they are, and in particular with respect to different spatial and sequence contexts, which I'll talk about right now. And so I'll highlight like two kind of stories that both illustrate slide-seq as well as these different contexts. And both of them are in this idea of like profiling actually the tumor microenvironment. And the first is this idea of sequencing T-cell sequences. So T-cells have receptors, right, which recognize antigens, and that T-cell receptor is different for different T-cells, and they can be clonally expanded, and understanding that T-cell receptor might allow you to also understand its relation to the T-cell clones with respect to the environment. And so first, let me talk about this. And the second is that in addition to RNA, we might also be interested in genomic DNA, in particular in the context of either in cancer for mutations, but maybe also in aging and somatic mutations. So I'll first talk about T cells. So there's some broad motivations for sequencing T cell receptors in the spatial context, in development and physiology. This is like a clearly spatially interesting process wherein the T cell repertoires have to move around, for example, in the thymus before developing, and they spatially distribute. And then also in pathology and autoimmunity, T cells, obviously different T cell clones are maybe differentially distributed in pathological conditions, and specific clones may be interacting with cancer cells or other cells to elicit immune responses. So first, I'll tell you about kind of this technology, an adaptation of the slide-seq technology that allows us to read out TCRs. And the idea is that now we can identify basically the clonotype of the T cell, which receptor it has, as well as its spatial location in the tissue, and then which T cell state it has. And this is really work that's done with in collaboration with Kathy Wu's group at the FCI. And there's a particular quirk challenge that we had to solve from a sequencing perspective for the sequence aficionados, which is just that all of our libraries are 3' libraries, and the TCRs are on the 5'. They're like, about 1,000 bases away from the 3'. And so we developed an approach, and in particular, the part of the TCRs that you care about, the CDR3 region, is on the 5'. And so we developed an approach to basically amplify that out using kind of a multiplex PCR strategy that allows us to amplify all the different V regions from the alpha and beta. So there's more than two, but in particular here we care about TCR alpha and beta from the receptor, and they have a diversity of V regions, and we can amplify that out from the 5' end and have a full-length library that includes the spatial barcode and then also the VDJ sequence. There's also another kind of cool technological trick that we did, which was like sequencing this with nanopore sequencing, because it's hard to actually sequence a 1,000 base pair library. But on top of all that, what you get at the end of the day is you get your gene expression from slide-seq, which is X, Y locations and genes. And then for each TCR, you also get its XY location, and then maybe also what type of cell it is where this gene expression is distinct. And so let's just look at some data to see what that means. So first we did some validation where we performed this sort of sequencing on OT1 mice, which are just like a genetically engineered mice with a specific T cell clone. So this is in the spleen, and here you can see like the histology of the tissue and then kind of a reconstruction of the cell types. And you can see that the cell type representation is underlined by different gene expressions. It's like not synthetic, but we're just plotting the gene expression in space. For example, marginal zones, red blood cells, and T cells. And this is just to orient you on the tissue, but then for these T cells, we can also look at the expression of from basically this fraction of the library that has the unique receptor sequence, like the alpha or the beta, and you can see the correlation of that to the capture of just those transcripts themselves. These are the constant regions, and 99% of these TCR variable sequences match this OT1 sequence. I mean, this mice only has the TCR, the OT1 sequence. But more importantly, we see high concordance between this dial-out library and then the underlying sequence locations. And the other thing is that we, what's actually the capture efficiency of this variable region? Well, depending on how many transcripts that each bead saw of the constant, we get higher and higher capture efficiency of the variable, but we estimate that we're probably getting something like 20% of the TCR transcripts can we assign one of these variable sequences. So there's a lot of room for improvement, but this is actually quite a challenging type of measurement. So we're getting the clonotype information and also the spatial information. So just in this example, right, like if we think about kind of a biological application, if we apply this sort of measurement within for example, cancer, how do T cells vary clones? Like as a function of what clonotype you are, where do you vary in your infiltration inside of the tumor? And so we applied this to a tumor specimen from a human. Actually, this is a secondary lung metastasis from a renal cell carcinoma. And so here is the H&E, and you can see basically this is the tumor region. There's an immune-rich boundary, and then this is a metastasis into the lung. And so we can actually identify these regions through just like, this is unbiased clustering of the cell type distributions. And the lung regions obviously have a lot of lung cells. And so we can segment that. And then here's this myeloid-rich boundary. And then here's the tumor cells. Actually, kind of this cancer is kind of very unique in the sense that in these mets that we often observe this myeloid-rich boundary of like monocytes and macrophages. Okay, so that's the cell type distribution. So where are actually also the T cells and the clonotypes? So here we did a very simple analysis, which we looked at basically for each TCR clone that we identified, is it enriched in the tumor going this way, or is it depleted? And relative to, basically, relative to all the other T cells. And you can see that for certain certain clones, for example, TCR4, it's kind of enriched in the tumor, and then other clones, it's depleted. But if you look at basically, so each one of these dots represents the location of one cell with a given TCR receptor clonotype, right? And for example, TCR8 is depleted. It actually has a lot more enriched in this boundary, and each one of these is their clonotype. If you compare this enrichment as you measure from the spatial measurement, this near single cell spatial measurement, this bulk fraction of the TCR clonotypes is not predictive at all of infiltration. So we also have paired bulk sequencing from the blood or from the same sample, and basically the relative amount expansion of these clonotypes is not indicative of how infiltrated they are. There's something about either the clonotype or the gene expression of the cells carrying that clonotype that's predictive of infiltration. And so we wish to examine that a little bit more. So ignoring all clonotypes, let's just look at T cells. There is this gene expression signature that's like poor response to immunotherapy. It's like an exhaustion signature. And it's from a paper from Nir Cohen's lab. And so if you just rank the cells in the T cells in the tumor by their expression of this gene expression signature, so they're either greater than the median expression or less than the median expression of this exhaustion signature, you can see, for example, that all of the cells that have really higher expression of this exhaustion signature are enriched at the boundary, and then the ones that are low in the exhaustion signature is enriched, more infiltrated in. And how does this break down by clonotype? Basically, almost for all clonotypes, decreased PRI expression is associated with deeper infiltration, but it's stronger in particular for the clones which have higher enrichment in the tumor. This suggests that there's something about the clone themselves but also there's a distribution of states within each clone that affects like basically how exhausted they are, whether they can infiltrate into the tumor. So there's this kind of this three-dimensional axis, right, where you have to be the right clone, and then you might also have to have the right gene expression states, and different clones might also have different gene expression states that affect their spatial localization. Okay, so switching gears a little bit to talk about, also in the context of tumors, capturing genomic DNA as well. And so here we were capturing T-cell receptor sequences from mRNA, but in many cases, for example, in the case of chromosomal number alterations or actual mutations, you might care about capturing genomic DNA in space. And we figured that there was a quick way to modify these beads instead of capturing poly A mRNA to also capture DNA. And so how we do that is we take the same bead arrays and we just change out the sequence from poly A to an adapter sequence. And this adapter, importantly, allows us to capture basically DNA that's been taggmented with TN5 transposase. TN5 transposase is like an enzyme that randomly inserts adapters into DNA. We can take a tissue section, fix it, and we can actually insert these adapters into the genomic DNA, and then we can ligate them locally to the bead oligo. So now instead of capturing mRNA, we're capturing random pieces of the genome. And the idea is that basically this sort of protocol is the same as the transcriptome. So on serial sections, you can capture DNA and then RNA, and you can register them together. You don't capture them exactly on the same tissue, but on just directly adjacent tissues. And the way to make the arrays is the same. So first we did some validation of this sort of method in mice. What we did was we took clones, mutant clones of tumor cells and injected them into mice so that they formed metastases in the liver. And we know actually what the CNV profile, these clones have known CNV profiles. And then we performed this sort of measurement. And here we've just binned, when you do this measurement, you get basically reads across the genome. And we just binned the reads across the genome in 1 megabase bins. And then we performed PCA on those 1 megabase bins. And so the first principal component of variation on those 1 megabase bins, you can see actually corresponds to tumor cells. So here's an adjacent immunofluorescence, and GMGA is marking the tumor cells. And you can actually see, if you just cluster it into two groups, that the tumor cells have known like chromosomal alterations that are marking them. And you can do this similarly with two different clones. Here we inject two different types cell lines that have independent CNVs. Now when you perform PCA on the genomic bins, you see that PC1 and 2 actually separate out the tumor and the different clones. And what is actually like marking those? Like if you look at the loadings on PCA, you can see that this first clone A has a chromosome 6 amplification here in a subset of chromosome 6, but chromosome 15 specifically marks the second clone, and then they both share this amplification in chromosome 19. So this is just in mouse data, but you can like identify spatially different clones through the DNA sequencing. It's like there's no RNA, you're just like clustering the counts of DNA to identify the localizations of clones. We also validated basically using the known CNVs from these that this is correct. Okay, so we can then apply this on human samples. And so here, we applied it on this colorectal adenocarcinoma from a human patient. And actually, here we performed H&E, and we performed basically both RNA and DNA slide-seq. And again, PC1 seems to separate out tumor versus normal tissue. And you can see that here. Here's the H&E, here's PC1. Here's MKI67 marking tumor cells, and then CD45 marking immune cells, which is infiltrating all around these like kind of lobules of tumor. But PC2 seems to separate a subclone. So this is kind of one tumor that has evolved in two different subclones. Actually, there's probably a couple more subclones, but at this resolution, there's two different subclones. And you can see that there is a MYC-driven tumor, so they all have actually chromosome 8q amplification. And then the subclone is actually marked by chromosome 20. A couple of different alterations, but one alteration, for example, is this chromosome 20 amplification. So these are all spatial plots. You're visualizing the spatial chromosomal number alterations here. Okay, so what can we do with this sort of information? What sort of analyses can we do? Well, one thing we did was we collected an adjacent section paired spatial RNA sequencing. This first sort of it. So here is slide RNA-seq, which is capturing mRNA from this section, and here we just clustered each cell by whether it's tumor, normal, or an immune cell. And you can register directly on an adjacent section this sort of DNA measurement where now we've labeled the clone information, whether it's clone 1 versus clone 2, or clone 2 has that, if you remember, that chromosome 20 alteration. And so one thing that a simple analysis we did was we'd like to understand how much of the gene expression in these tumor cells is a function of its intrinsic chromosomal number alteration versus the extrinsic environment, who its neighbors are. And one proxy for who its neighbors are we just quickly did was basically tumor cell density. So as a function of how many tumor cells locally you saw. And this is just a heat map of that density. You can see that these different lobes have kind of varying densities. And so the intrinsic here is the clonal assignment, and the extrinsic is something like the neighborhood expression. And so we did a regression to see how much of gene expression variance can you explain by these two variables. So that's the results. Here are the results of this regression. So this is kind of a fancy plot, but it's easy to explain. So each dot here is a gene, and we regress how much of the variance in that gene can you explain on this axis, which is tumor cell density, versus this axis, which is what clone it belongs to. So some of the variance can be explained by if you're in clone 1 versus clone 2, and some of the variance can be explained by some local environmental factor. And a lot of the variance can't be explained by either of those things because the majority of genes are in the middle, right? So maybe they're not affected by either clone. But for example, there are some genes like PLAG1, for example, where almost all of its variance is explained by which subclone it's associated with. Just for reference, here's clone 2 versus clone 1. It's just very highly expressed only in clone 2. Maybe MCM7 is another example, but PM3D is the opposite. And then there are some other genes which are not at all explained by subclone, but they're only explained by the local density of tumor cells. For example, LGAL-S3 and PROM1. These happen to be kind of adhesion or extracellular matrix proteins. And actually, if you just run a gene ontology analysis, you can see that the subclone-associated gene sets tend to actually be MYC target genes because one of the subclones has a higher amplification of MYC than the other. That kind of makes sense. And then the density-associated genes happen to be kind of cell adhesion and coherent genes. So there's some program that the cells are running that's independent of what clone it is, but it seems to be affecting its local environment, how locally dense the tumor cells are. For example, here are all the genes that are cell adhesion in the cell adhesion and density associated gene sets. A lot of them are enriched on this upper half, which is in the local cell density. And so, so I'm coming to the first half of the talk. So I'm just going to summarize a couple things about SlideSeq. It's an experimental platform for high-resolution spatial transcriptomics. It allows for example, studying the relationship between spatial locations, the transcriptomes of a cell, And then when we do TCR sequencing, also the TCR clonotypes. And I think one thing that we're very interested in is understanding cell intrinsic, like environmental, versus cell extrinsic, like genotype or clonotype. So in like these two examples, we looked at cell intrinsic, which was like what TCR clone you are or what tumor clone you are, versus like extrinsic, which is like where you are in the tissue, right? And so that's something that we've been trying to tackle with these analyses. Now, so these are some technologies that we developed, a couple years ago. But one challenge that we always had with this sort of like spatial capture technology is that actually like it's not true single cell resolution. It's 10 micron beads, and mammalian cells are like 5 to 10 microns. So some of the times you capture one cell, and some of the times you capture two cells. And we came up with a lot of like clever computational analyses, like methods to deal with that exact problem, like this problem of mixing, how to do differential expression in the context of mixing, and I think that's valuable because there's a lot of new technologies on that are using this sort of spatial capture method. But we really like in another part of the lab's work have been trying to use these technologies for like tissue atlases, like collecting comprehensive like both collections and organizations of organs, tissues, and in particular in the mouse brain, for example. And we actually did a study where we did single cell sequencing of 6 million single cell profiles across the entire mammalian brain, and then on a single mouse brain, took 101 serial sections and reconstructed like a 3D atlas of like where those cell types are in every section. But I think this challenge that you always have to do single cell, and then you have to come up with some clever computational approaches to register the single cell data with your spatial data was bothering us. And so we wanted to come up with an approach where we wanted to just like get true single cell data, and then at the end of the day know where the cells were. And so some folks in the lab started working on this, and this is work that's been recently published. It's really led by Andy and Jackson, or in my lab, and then Naeem, who's in Evan Micasso's lab, who we've been working on these technologies together. And so the idea actually was quite simple, which was, in SlideSeq, we are trying to capture the mRNAs and barcode them with these beads. And we basically like inverted the problem. It's like maybe we should, instead of trying to barcode the mRNAs, let's try to barcode the cells with the beads, and then put the cells into single cell methods. And there's a couple advantages to that, which I'll explain. But the idea is the same. You have the same beads, which I already told you about, these bead arrays that are barcoded, have lots of oligos on them. And now instead of thinking as the beads as pixels, we can think of them as satellites, where they're releasing information about the XY location. We put the tissue onto these bead arrays, and then the idea is called slide tags. We're trying to tag the nuclei with these satellite sequences. So the tissue sitting on top of these beads, we can photocleave the oligos. We can use like this like light, 365 nanometer UV light that photocleaves these oligos off the beads. And they diffuse into now the nuclei instead of the RNA diffusing into the beads. And then this process happens very quickly in about 7 minutes. And we don't actually know the mechanism by which these oligos associate with the nuclei. We just empirically observe that the nuclei are very sticky to oligos. And that seems to make the protocol work. That question comes up a lot. I just want to preempt that. And then we isolate the nuclei and we put them into single cell profiling technologies. Here we're doing like very established, all the data that I'll show you was going to be with 10x basically, but there's a lot of different single cell technologies out there. Today, 10x happens to be one that's relatively easy to use. And when you put that there, when you put these things in the droplets, what's happening is that inside your nucleus you have your mRNAs, but you also have your spatial barcode, which tells you what, which beads it was next to, and then they get captured by inside this droplet by another set of beads that's part of the 10x platform. And at the end of the day, you can associate the same cell barcode with a spatial barcode library and a gene expression library. So, I mean, what does this actually look like? So I'll just highlight this by some example data in the Malcific campus, which we often like to look at for technology development. And the idea is simple. You do this sort of tagging procedure, you do single nucleus RNA-seq, and what you end up with after you do that is like a matrix of cells by genes. And oftentimes, people will then go and do some clustering, like dimensionality reduction and clustering with these genes, and you end up with something that looks like this, like a UMAP. And we don't have to really talk about this very much, except for to say that each one of these dots is a single cell, and we've clustered them in these clusters by like what cell type they are. You'll see that in a second. The cells that are in a given cluster together are like similar in gene expression. But importantly, if you look at individual cells, they've also captured like some of these satellite sequences about where they're coming from. And so like this green cell, right, captured a bunch of information, satellite information from x equals 2000 and y equals 1500 or 1200 or something. And you can just like fit those locations. And it turns out you can fit those locations quite accurately. And in fact, we came up, then you can get the XY coordinates for each one of these cells. And in fact, we can do that. So like we can reconstruct, not to reconstruct, we're exactly placing these cells from where they came from. And the key is underlying each one of these cells is actually the gene expression from those cells. So you can then like plot expressions of marker genes, for example. So here is an in situ hybridization, and here is like the marker gene expression from those same cells. And so not only do you get the cell type organization, you also get the gene expression profiles. And so why do this instead of just capturing the mRNA directly? Well, I think there's like some really key advantages. One is that the data quality is like indistinguishable from just doing normal single nucleus RNA-seq or single cell RNA-seq in both cell type proportions. So this is on adjacent sections performing just normal single nucleus RNA-seq versus this tagging approach. And like cell type proportions and the counts per cell, and also in the average gene expression profiles. They're like exactly the same. So you like sacrifice nothing in terms of the data, but now you get the spatial localization. The spatial resolution we characterize from like many different ways, but one way, for example, is looking at like an adjacent section and seeing like basically the width or size of histological features, and we think that we're placing single cell locations with at least 10 micron resolution. That's about the size of the bead. So you get single cell profiles, but you know where those single cells profiles are coming from with about 10 micron resolution. And then, so what's one important metric is that we don't sample every nuclei from the tissue. We sample about 15% of the nuclei. And that's because you lose some nuclei in nuclei expression and you lose some nuclei in 10x. And we've been improving this quite a lot in other versions of the protocol, but I think 15% of the nuclei is quite good, actually, because for this other reason, in the sense that the data quality is much, much higher per nucleus compared to other spatial methods, including slide-seq. You get about an order of magnitude more molecules per cell compared to the spatial capture approach. And here are some other comparison technologies. We don't have to talk about that. But I think the main important thing is that the data is true single cell resolution. And that actually is really important for analysis, because here is like taking your data and running, this is 4 data sets from the Malcific campus, and we ran principal component analysis. So like principal component analysis breaks down this matrix of gene expression by components which are ranked by how much variance they have. And so if you just look at the standard deviation of the different components across all your cells, you can see that they're ranked. But importantly, you see that in an approach with true single cell resolution, you get many, many more components out here that have like variants. And that's because when you mix the cells, you know, you lose like this fine-grained molecular information. You can recover like global cell types, right? Because like a neuron is going to be very different from an astrocyte, so that's like PC1 or whatever, or PC2. But you know, states within those cells are going to be mixed, and so there's just like a lot more information in the single nuclei or single cell data. And the other thing that I think is important is that basically we've converted, like, this method is like an upfront step where you can convert any existing single cell technology into a spatial technology. So, you know, people have spent a lot of effort developing a bunch of single cell technologies for capturing mRNA, DNA, full-length RNA, TCR sequences, and also like epigenomics, like chromatin accessibility, where you can like do this sort of thing up front, and then you can isolate the nuclei and you can get the spatial locations of all of those technologies as well. And so that's like the two reasons why we are pretty excited about tags. And we published a paper about this recently where we demonstrated many different ways you could use it, like in the human cortex, in the mouse, in human tonsils. But I'll just highlight like one example in this metastatic melanoma case that really highlights like a couple qualities. One, the data quality, and two, the fact that you can just integrate a lot of these different existing single cell analysis and experimental approaches. Because we do single nucleus RNA-seq, ATAC-seq, as well as CNV inference. And so we performed this sort of slide tags on this like metastatic melanoma sample, and this is a metastasis in the lymph node. And here is actually this is 3 millimeters in diameter, like an H&E image. And we noticed in the H&E that there was kind of like these two lobes of tumor cells separated by this fibrous stroma. And when we performed this sort of single nucleus RNA sequencing, you can resolve very clearly the different types of cells that are in here. Actually, there's quite a lot of molecular diversity inside of the T cell population as well as inside of tumor cells. And you can place all of these cells back into space. It's a little bit hard to see here with all these different colors, so let's just focus on the tumor cells for a second. So in the tumor cells, you see that there are these transcriptionally distinct clusters, right? They separate out in the UMAP, but they also are spatially distinct. Actually, tumor 1 and 2 populations separate across these two lobes, across this fibrous stroma, but also 1B has this like local clustering in space as well, which is quite interesting. Not only that, we can run something called infer-CNV. This is like looking at the chromosomal number alterations through the RNA-seq data. And this is something that you can really only do with single nucleus or single cell resolution data. And you can see that in the normal cells, there's white represents no alterations, red represents a gain, and then blue represents a loss. And so you can see some shared chromosomal alterations because it's the same tumor. There's like two different evolutionary clones or related clones of it, right? And so they'll have this chromosome 7 gain, 9 loss, and you can actually see that they're quite similar across a lot of it, like 13. But there's some private stuff, right? In particular, there's these private losses of chromosome 6 that's special to 1. Actually, quite a lot of stuff that's unique to 1, right? This loss in 3, as well as 4. And so there's these two like genetically distinct, transcriptionally distinct subclones that are also like spatially segregated. And since this is spatial data, we can look at like what's different about the spatial environments, the microenvironments of these tumor populations. So one simple analysis you can do is you could just look at the enrichment or depletion of other neighboring cell types. So here, red means a cell type is enriched near tumor 1 or 2, and blue means it's depleted. And for example, you see that CD8 T cells are strongly enriched in tumor 2, but strongly depleted in the tumor 1 population. In fact, most immune cells are depleted from tumor 1 CD8 Ts are enriched in tumor 2. And so what's actually different about these T cells that are in the tumor 2 neighborhood? Well, T cells in tumor 2 have a much more cytotoxic phenotype. You can see they have granzyme B, and then there's some immunosuppressive markers here in tumor 1. So similarly now, we can look at TCRs. We can use TCR sequencing in the same way that I just described with this targeted RhTCR sequencing, and we can recover the TCR clones from more than half of the T cells in the sample. And you can see there's a relatively large clone here, and if we plot the localization T cells, you can see that this clone is highlighted in purple. So the T cells, these are almost all CD8 T cells. The T cells that have this clonotype is in purple, and then all other T cells are in gray. And so you can see like this particular clonotype has this very strong bias for tumor 2 versus tumor 1. And that's actually, this clonotype is actually driving the enrichment of T cells in tumor 1. And this is also the more cytotoxic clonotype. And so there's like a question of like, why is this happening? And basically there's two hypotheses. One is that we see this inferred copy number loss on chromosome 6. This suggests that maybe there's this HSLA locus is on chromosome 6 there, and there could be there's like both a loss in endogenous antigen presentation machinery, but you could also have just like a loss of heterozygosity of HLA, and that could lead to basically some antigen that's being presented by tumor 1 not being able to be presented for that T cell clonotype that the antigen is being represented. The second thing which we were interested in looking at is basically like these melanomas have this very particular state transition that they often undergo, which is this deep differentiation from this melanocytic state to a more mesenchymal-like or neurocrest-like state. And we thought that this might be true when we looked at our RNA data. And so we carried out a joint experiment with both single nucleus RNA-seq and single nucleus ATAC-seq. So this is done on a multi-omic platform where in the same cells you can collect the chromatin accessibility as well as the RNA. And what we did here is that we can now still do this sort of tagging approach, and you can put the cells in space, and you get a very similar clustering, and also, again, the separation between tumor 2 and tumor 1, as well as the infiltration of immune cells. When we look at the epigenomic differences between these tumor cell populations, we see actually quite a lot of markers that previously been identified for this like dedifferentiation, including like TNC and PLCB4. Here's like expression of TNC, just a track, as well as this is an accessibility track, and here's the RNA expression. So they're both different in accessibility and gene expression, and you can see that many cells in tumor 1 have this chromatin accessibility of TNC. And then if you just generate a mesenchymal-like score, you can see that tumor 1 is much higher in this mesenchymal-like score, and in particular, they also lose mLNA and PML, which are endogenous antigens that's commonly associated with T cell response. So that's interesting. If you look at TF motifs, this is kind of a very interesting analysis in the sense that you can look at TF motifs and look at the correlation with this mesenchymal score. So there are a lot of TFs that are associated with this mesenchymal-like state that's over on this axis, and then the TFs that are associated with the melanocyte state are on this axis. And MITF is this master transcriptional regulator of melanocyte state, so it's over here, that's good. But there's a lot of like FOS, Jun, and IRF signaling that's correlated with the mesenchymal-like state that's maybe driving this. That's also been something that's been relatively well characterized in the literature. So, but on the y-axis, I think there's something quite interesting, which is we can look at basically the clustering of these states in space, and you see that a lot of these IRF signaling is spatially clustered. So, like in the tumor cells that are in tumor 1, they have like local clustering of upregulation of these these TF motifs, and these are also happen to be some response to inflammatory signaling. So there could be some local inflammation that's actually driving this dedifferentiation. Anyway, so I think we're generally, again, this is another example of like looking at differences between the intrinsic like environmental factors versus extrinsic environmental factors versus like the intrinsic tumor cell clone or T cell clone. And so we're generally interested in that. So just to summarize this part 2 for slide tags, we're pretty excited about this because it's like a way to just do single cell and then get where your spatial localizations of single cells are, and it plugs into existing single cell workflows. And we're working quite a lot on adapting a lot of genomic and epigenomic measurements because we think this is an area where there's not been very much advancement in the field for spatial. And then we're also thinking about spatial perturbations to understand cell type functions. And so in general, like the lab is interested in taking measurements in the tissue context, where here we've been developing a lot of approaches to look at the transcriptome, epigenome, and genome, but we're interested in using these sorts of things to look at interactions between cells, lineage, dynamics, as well as basically perturbations that we can do to understand more than just correlation, but also causality in these systems. So I think I'm basically out of time, so I'm gonna end here, and I'll quickly skip these slides, and I'll thank the people who actually did the work. So Andy, Naeem, and Jackson really led the slide tags work, really in a close collaboration with Evan, and then Kathy and Sophia. Sophia led really the IMU TCR work in collaboration with Kathy's lab. Thank you, and I'll take any questions.
So in your slide tags methodology, you've got a big increase in the amount of data you're able to recover from the single cell sequencing. Could you tell us more about why you think you're able to recover more data using the slide tag method versus, straightforward single cell sequencing?
So the slide tag method gives you we are getting the same quality of data as normal single nucleus sequencing. It's just much, much better than slide-seq, which is capturing it on the arrays. And I think it's just because the local enzymatics of like diffusion or capture onto the arrays is just not nearly as efficient as doing it in the droplet, where you get to like lyse the cell. And I think actually like the droplet-based approaches are quite high quality in data these days, and so it's quite exciting.
Very interesting. Is there, I assume, a lower limit to the size of the beads?
Yeah.
Why can't you go into much smaller beads?
Yeah, no, I think that's a good question. Actually, that also is always asked. And I think you can go to smaller beads. In fact, we've also made a little bit smaller beads. But one thing is that basically the mRNA molecules per area is the same. So when you go half the size of the bead, each bead is collecting 1/4 the information. And so what tends to happen is that then you have to re-aggregate the beads for analysis. If you go to 2 micron beads, for example, because that's going to be 1/25 the data per bead. And so we haven't been going down that route because of the computational, like the data quality. Like we think 10 microns is great for data quality. And then I said, we just saw like if you do the single cell version, you can get like an order of magnitude more mRNAs per cell, per location. So we're really excited about that. Okay, 2 more. Yeah.
How do you prevent in the slide tag where after you photo cleave in the microscope, how do you prevent 2 sets of adjacent tags, diffusing and mixing in a boundary?
you do, okay, so that's exactly a great question, and I will show you. Basically, we are seeing that in the data. We see that each cell, so it's hard to see here, but each little diamond is a unique bead. And so this cell has picked up like a cluster, like it's picking up oligos from like 20 or 30 different beads. But that's actually helpful because we're then fitting, it has this classic diffusion profile. You can actually fit the location of that cell to the centroid of this diffusion profile. You can see that here. And we did some modeling of that, that basically that actually improves the performance. We rely on it picking up multiple nearby beads so we can actually spatially localize it.
So timing is very important. You can't let it diffuse too long.
Yeah, you can't let it diffuse too long. You can't let it diffuse too long, that's correct. And so we do it for like 5 minutes, basically. I don't know if it's that sensitive from 5 to 20 or something, but it is, if you wait an hour or something, you will not get anything from it.
Yeah, kind of like a follow-up to that. Can you deliver all the tags for the same type of analysis you want to do, or do you have to do them on the previous first part of the talk for the genomic analysis? You have to deliver a different set of tags on a separate section, or can you flood tags for ATAC-seq, genomic, copy number variation, and RNA-seq in the same place? Yeah,
that's what we want. We're doing that. We currently did tags for our ATAC-seq, copy number, and RNA in the same sample. The only caveat is that the copy number is converted through RNA or through the ATAC-seq. We're working on currently doing true genome sequencing, right? Low coverage genome sequencing, but still sequencing the genome in combination with RNA, which is still a challenge for single cell methods. But in the same measurement, same cells. All of those analyses were in the exact same cells.
Same section.
Yeah, same section.
I have a question about expanding this on many samples. Super exciting technology, but you're showing us kind of candidate samples. What happens when you have 12 samples and you're trying to do this? How different is
that's a great question. So I showed vignettes for every single one. We've done that a lot, I think, for SlideSeq. We did 100 serial sections from the same sample. And we are starting now to do quite a lot of collections for tags. We ran about 20 samples of melanoma in the last 2 months with tags. And I think the takeaway is we had to learn a lot of lessons about how to do it in the beginning. And then now I think we kind of know how to do it. And so in the beginning we had to work out a lot of quirks. And then recently in these 20 samples, after we worked out all the quirks, basically we got very high quality data from about 80 to 90% of them. And then there was 1 to 2 samples that probably we can't do.
But it's comparable.
Yeah, I think it's comparable in the robustness. Now, I think, it's just we're learning the lessons about how to do it. It's not going to, we can't do it on every sample when we scale it, but there's some sample to sample. I mean, they're all human samples, so I think doing it on 80% of the samples successfully as well is pretty good.
Was there batch issues between samples that you struggle with? Did you do the slide tag, not necessarily the single?
Yeah, with slide tags, the batch, it's the same sort of batch effects that you normally see with single cell or single nucleus, and we do see batch effects, and you try to correct for those, but it's not any more or less than just running a large cohort of single nuclei. Pretty reasonable batch effects, I think, actually.
I have a couple of questions, but let's just start with one. I think just looking at the PCR clones that you showed, that was just in one single network. You basically showed that the clones are like either in or out.
Yeah.
If you look like at a different cancer clone, do you see the same? Is that the same? Is it intrinsic or is it related to the cancer clone?
Yeah, we don't. Yeah, exactly. That's a good question. For that sample, we don't have a lot of other clones, and so that's why we started this like larger. In this collection of 20 melanoma samples, we actually have TCR as well. And so that's like one question where we also observe consistently like CMV, like lots of different CMV clones that are spatially separate, which is like kind of an interesting. And so we're trying to ask like if we see consistently like different clones, TCR clones that are associated with different tumor clones. The question is like, I think maybe you're trying to get at is maybe we could sample like multiple MET, like primaries and METs from the same patient and see if like the clones that are associated with one region are associated with the same.
Right.
I think it's really just a sample. It depends on having the right samples, and we don't currently have that actually.
And a more theological question. I think you compared to Xenium, which is obviously in situ based, is supposedly very sensitive.
Yeah.
And you basically showed you have a higher number of UMIs, but that might reflect your complexity that's higher, because you're unbiased. But is it the same in terms of sensitivity?
I think it's very similar in terms of sensitivity as Xenium, actually, because if you look at the Xenium measurements, they've compared the sensitivity of Xenium to their single cell products, and it's like maybe quite similar, actually. And then we're just using their single cell products, so we aren't. I don't think the sensitivity is coming from us; it's just coming from TEDx. Droplet-based RNA sequence is very good. But I do think there's this one thing about the Xenium is that if you look at this metric, okay, so like there's two things about this. One is that like there's less genes, so maybe it's not exactly a fair comparison there because it's only like 300 genes or something. So, but they chose the genes to capture some of the variants in the sample, right? But I think part of this thing that's affecting the PCs there is also like the segmentation. The segmentation leads to like mixing between cells in the same way that pixels lead to mixing between cells, but to a lesser extent, and that's also a thing that you see in the Xenium data.
Maybe one last question, just out of interest, like you do the copy number variation on the DNA.
Yeah.
That's obviously on the 1 megabase, which is pretty big.
Yeah, pretty
what
big.
Is the actual resolution? Can you do anything at a smaller scale?
I think basically both on the RNA side and also the slide DNA side, the CMV resolution right now is like at about a megabase. That's why we're interested in doing this sort of like true single cell DNA profiling, because there you can get like 50 kilobase bins or 100, about 10 times better.
Yeah, 324.
That's just the GSI. Yeah.
Yeah.